Hello children. Today we'll be continuing with the reproductive health and we are in the last topic of this chapter, that is assisted reproductive technologies. So what do I mean by assisted reproductive technology? So what do I mean by assisted reproductive technologies? It means all the medical procedures which are used to address to the problem of infertility. And what is infertility? Infertility means incapability to bear a child even after having 1 year of unprotected coitus. This assisted reproductive technology is also denoted with a short form, and that is ART. So when we say ART, we actually refer to assisted reproductive technologies. So the assisted reproductive technologies which we will study about today and which is there in your syllabus, they are 5 types. The first one is in vitro fertilization, which can also be called as test tube baby. The short form of in vitro fertilization is IVF-ET. The second one is gamete intrafallopian transfer. The short form is GIFT. The third one is zygote intrafallopian transfer. The short form is ZIFT. The fourth one is intracytoplasmic sperm injection. The short form is ICSI. And the last one and the fifth one is testicular sperm extraction, and the full form is TESE. Clear? So now we will get into the details of how these procedures take place. So we are starting with the first one, which is in vitro fertilization or the test tube baby. When I say in vitro, I mean the fertilization will take place outside the body. In vivo means inside the body. In vitro means outside the body. So when the fertilization is taking place outside the body, it means it is taking place in a laboratory dish or maybe in a test tube. Clear? So in vitro fertilization is the joining of women's egg and a man's sperm in the laboratory dish. Okay, the fertilization takes place in the laboratory dish, which is the attachment or the sperm penetrating the egg. So let us see what are the steps that take place in vitro fertilization. The first step is known as stimulation or superovulation. When I say superovulation, I obviously mean this is taking place in women's body. So, to induce superovulation, medicines called fertility drugs are given to women to boost their egg production or their gamete production. We know that every month there is only one of the ovaries synthesizing one of the eggs. Alternately, the two ovaries take part in the cycle. But here, when in vitro fertilization is supposed to take place, drugs are given to the women so that egg production is boosted up. Normally, a woman produces 1 egg per month, but when drugs are given, ovaries produce several eggs. And when this superovulation is done, a regular ultrasound is done so as to check the ovaries and the hormonal levels inside the female's body. Once superovulation is done, egg retrieval procedure, which is the step 2, is performed. What is done? A minor surgery is done to remove eggs from the woman's body, because now this egg will be required for fertilization. And just because it is in vitro, so fertilization will take place outside the body, so the eggs need to come out of the body, and it will only come out by a minor surgery. Here, the minor surgery is known as follicular aspiration. So from the follicles, the egg will be taken out. Step 3. Step 3 is the insemination. Insemination means when the man's sperm will be placed together with the best quality eggs. And whenever there will be mixing of the sperm and the egg, that will be known as insemination. The sperm will enter the egg few hours after insemination, when it will get some controlled conditions in its environment. Now here I have mentioned one point, that if the doctor thinks the chance of fertilization is very low, the sperm is directly injected into the egg's cytoplasm. So basically in vitro fertilization, what is done? The egg and the sperm are placed together in a laboratory dish in some environmentally controlled chambers, and the sperm and the egg will mix with each other, and the egg will be fertilized by the sperm itself. But in case fertilization is not taking place itself, then the doctors, they inject the sperm directly into the cytoplasm of the egg. Coming to step 4, it is the embryo culture. So when the fertilized egg, we know it actually starts dividing for its development, and it becomes embryo. Right? So when the division will take place, the laboratory staffs will regularly check the embryo to see if it is properly growing or not. And within 5 days, a normal embryo will have several cells which will be actively dividing. The last step is the embryo transfer. So embryo will be placed into the woman's womb for 3 to 5 days after the fertilization. The doctor inserts the embryo with the help of a catheter into the womb of the woman, and the embryo will implant itself into the lining of the womb or the uterus, and the pregnancy will result from there. Right? So this is how a woman will conceive and will implant the embryo into the lining of the womb, which will result into pregnancy. Clear? Now here some additional information, like more than one embryo may be placed into the womb at the same time. That will lead to twins, triplets or more. And that completely depends on certain factors which will be decided by the doctors. Coming to the second type of assisted reproductive technology, and that is gamete intrafallopian transfer. Now when I say intrafallopian, I mean inside the fallopian tube. What will be transferred? It is the gamete which will be transferred. So in this technique, fertilization will actually take place inside the body of the female. The sperm will be injected into the fallopian tube of the female with the help of a laparoscopy, and then fertilization will take place as it takes place in the ampular region of the fallopian tube, and then further development will occur, which will be in vivo. That means inside the body of the female. The third one is zygote intrafallopian transfer. So this is the zygote will be transferred inside the fallopian tube. So that means the fertilization will take place in vitro. The sperm and the ovum will mix, or the ovum will fertilize in the laboratory. It will get proper conditions. It will start, and once it fuses and it forms zygote, the zygote will be taken and placed into the fallopian tube. Right? And then the further development process will take place inside the fallopian tube. And here it is said it's a better technique as fertilization is confirmed here. Which means that because fertilization is taking place in the laboratory, so it's a confirmed process. But in gamete intrafallopian transfer, the sperm is injected into the fallopian tube, which might not fertilize the ovum. The next one is the intracytoplasmic sperm injection. From the name we can understand, it is inside the cytoplasm, the sperm is injected. So this is also an in vitro technical method. While we were doing in vitro fertilization, I told you the sperm is directly injected within the cytoplasm of the ovum with the help of a needle. So that means fertilization is taking place. Now the zygote which will be formed, or after the embryo is formed, that is taken and either zift or gift. Either zift is done or it is waited till the embryo stage and then placed in the womb or the uterus. And the last one is the testicular sperm extraction. Now this is for men who are unable to produce sperm by ejaculation. Okay. Men who don't have sperm in their semen, or men who have absence of vas deferens from birth, or there has been a non-reconstructed vasectomy. That means the vas deferens is not complete. So in that case, what is done? A small portion of the tissue is taken from the testicles under local anesthesia, and sperm cells, viable sperm cells, are extracted from the tissue. And then the sperms are taken and again ICSI is formed. So just because here the man is unable to produce sperms, so the sperm is directly taken from the tissue of the testicles, and then it performs ICSI to initiate the further processes. Clear? So this was all about the assisted reproductive technologies. Thank you.